Welcome all. In today's lecture, I will talk about the recombinant protein expression by using bacterial expression system. We have different types of expression system which we can use for this purpose. For instance, we have bacterial cells, we have yeast cells, we have mammalian cells, we have insect cells. So they can be used for expression of our protein of interest. However, in this particular talk, I will discuss about bacterial expression system, or how we can use bacterial cells to express or synthesize our protein of interest. Now, this whole procedure of bacterial expression system, I have divided this whole process into three main parts, or three main sections. So number one is the cloning of expression vector. So the first step is that we should have many copies of the expression vector. So we have to clone our expression vector, the plasmid. When I say expression vector, that means the plasmid having our gene of interest. So how do we do this cloning? We use a particular strain of bacteria that is known as DH5 alpha, which is E. coli bacterial strain, and it is used specifically for this purpose. So with this cloning, we will have many copies of our expression vector. So we can use certain amount to do the transformation of protein expression bacterial cells, and we can we can store certain amount of the expression vector in a minus 80 degrees Celsius for later use. But if we already have a substantial amount of the expression vector, then this whole part can be avoided, and then we can start directly with the recombinant protein expression. Now, the second part of this whole process is the recombinant protein expression. And what does it involve? Number one is the protein expression. So we transform bacterial cells, a particular bacterial cells genetically engineered for expression purposes. We express our protein of interest, and once we are done with the expression, then we have to extract that protein of interest from these bacterial cells. So after extraction, what we are going to do. Now, one important thing here in this expression is that the expressed proteins are in the form of inclusion bodies. Now, what are inclusion bodies? Inclusion bodies are basically protein aggregates which are synthesized. The bacterial cells synthesize our protein of interest as inclusion bodies, and they are protein aggregates. They accumulate inside bacterial cells in a form of patches scattered all around the bacterial cells. So we have to extract these protein aggregates, which are as they are aggregates, so they are unfolded, they are not folded properly. So we collect and extract these protein aggregates or inclusion bodies, and then we have to unfold them completely. So for unfolding, we use a highly denaturing conditions. So why we do this unfolding is because in a bacterial system, as the inclusion bodies, they are synthesized in the form of protein aggregates, so they are not folded. We have to refold them in a lab environment. However, for proper refolding, we have to unfold them first. So we have to make them completely denature, and then we have to use a refold buffer to refold them. So these two steps, the protein unfolding and refolding steps, they are performed outside in a lab environment. However, the protein expression that takes place inside the bacterial cells. Now, the third main part of this whole process is the purification of recombinant protein, which is the ultimate goal and the last step as well. So the idea is to have to completely to purify a pure form, a mature form of the protein of interest. I have used the word active as well. So for instance, if we are dealing with enzymes, like if we are dealing with proteases, then we should have a pure form, pure active form of the protein as well. For purification purpose, we use different strategies depending upon the properties of the protein. So I will talk about that later in this lecture. Okay, so let's start from the first part, which is the cloning of expression vector. And I have tried to mention the main steps involved in this part in the form of a flowchart, because the idea is that once we have a complete picture of the whole process, right from the beginning of the expression vector till the purification of a protein, then I can discuss individual steps in more detail in subsequent lectures. So let's start with the first step in the first part, which is transformation of the competent cells. So the first part is the transformation of the competent cells, known as DH5 alpha, and these are specifically designed for cloning purposes. I have just mentioned two of the main features these DH5 alpha cells have. Number one is the mutation in REC-A gene, which ensures high insert stability, and there will be no heterologous recombination. So that means that once these expression vectors they get into bacterial cells, they will stay intact. They will not attach or recombine with the bacterial genome. And the second feature is that these DH5 alpha cells, they lack some endonucleases, which otherwise digest the plasmid. So once we have, these DH5 alpha cells and a mixture of DH5 alpha cells and expression vector, then the next step is to, do the transformation. So how do we do this transformation? Is basically by a simple treatment. We call that as heat shock treatment. So what we do in this heat shock treatment is that we keep this mixture of DH5 alpha cells and expression vector for 5 to 10 minutes on ice, and then we move that to 45 degrees Celsius and keep that for 30 seconds. And after that, we put that mixture back on ice for 2 minutes. So this sudden change of temperature from ice to 42 degrees centigrade is involved in this treatment. And then we inoculate that mixture with a liquid broth and let that incubate at 37 degrees Celsius for 45 minutes to grow these cells. The next step is to spread that bacterial culture on agar plates and leave that overnight at 37 degrees Celsius. So that is followed by the isolation of well-isolated colony, the picking of well-isolated colony and its growth in LB broth to make a bacterial culture. I'd like to mention, I have used the word competent. So competent cells are those that have the ability to take up foreign DNA from the surrounding. And the cells are made competent by a particular procedure. So competency is basically this ability of the cells to take up any foreign DNA from the surrounding. Okay, as I mentioned in my previous slide, that we pick a well-isolated colony and let these cells grow in a liquid media. So with that bacterial cells, with the growth of bacterial cells, our expression vector will also replicate, and we will have many copies inside bacterial cells. So after a certain time period, we just stop the growth of bacterial cells, and then it is followed by the extraction of expression vectors. Okay, so for the isolation or extraction of the expression vector, we use different preparation protocols, but that ultimately depends on how much particular concentration or amount of expression vector we are interested in at the end of the isolation preparation protocols. For instance, if we are interested in 20 microgram of expression vector, we follow mini prep protocol. If we are interested in 20 microgram of the expression vector, then we follow midi prep protocol. And if we are interested in 500 microgram of the expression vector or a plasmid having our gene of interest, then we follow the maxi prep protocol. So these all steps are almost similar with minor variations among these three different preparation protocols. So if we follow the mini prep protocol, at the end we get 20 microgram, approximately 20 microgram of the plasmid DNA. Now the next step is, once we have this plasmid DNA, our expression vector, so it means that we have cloned the expression vector. Now the next step is the expression of the desired gene in a expression vector. Now why we do this sequencing of the desired gene? So in molecular biology laboratories, transferring or shifting of gene of interest from one vector to another vector is a common practice. For instance, if bacterial system for protein expression for any particular gene is not working, then scientists use restriction in nucleases to cut that gene of interest from bacterial expression plasmid and ligate that to another vector, for instance in a yeast expression vector. So if we have a new expression vector, then it is always recommended that we do the sequencing of our gene of interest in that new expression vector, just to make sure that the sequence of our gene during this transfer from one vector to another vector is not disturbed. And once we make sure that the sequence of our gene of interest is not disturbed, it is perfectly fine, that means that we have cloned the expression vector, and now we have many copies of our expression vector. So the next step is to transform, do the transformation of a particular strain of bacteria that we will use for expression purpose, and that is the transformation of the BL21 bacterial strain. Okay, so this is the, as I mentioned before, that for transformation we use, this is a new strain, another strain BL21, which we transform by using the expression vector which we have cloned in our previous section. So we transform these bacterial strain, their particularly designed for expression purposes. They've got different features, very important features for this purpose, but I have just mentioned two of them. One is the it lacks these cells lack some proteases, so which ultimately proteases, you know very well that they degrade or digest proteins. So if they lack proteases, it means that our protein of interest will not be digested, will not be cleaved by these proteases. And the other important feature is that these cells have a mechanism through which we can induce them. So induce mean that we have to give these cells a sort of a signal so that they can start synthesizing our protein of interest when we want these bacterial cells to start synthesize that protein of interest. So this signal is given by IPTG. So if we are growing bacterial cells, transform bacterial cells, and after a certain time period to achieve a particular density or optical density of that of those when these growing cells attain a certain optical density, then we add IPTG. So it is a sort of a signals that will initiate these cells to start synthesizing our protein of interest. I will discuss this whole procedure in detail in my subsequent lectures. As I mentioned before, that the proteins synthesized in these cells, they are synthesized in the form of inclusion bodies. So inclusion bodies, they are protein aggregates, and they are synthesized and aggregated inside bacterial cells, scattered inside bacterial cells in the form of patches. So once the synthesis is complete, then we stop the growth of these cells, and then we collect these inclusion bodies. Okay, by the way, I didn't mention different steps involved in the transformation till we reach this point. So I have mentioned in detail that how this transformation takes place. So we again, when we transform these BL21, which is a bacterial strain used particularly for expression purpose, so we follow similar steps as we did in our previous section, that is the when we transform DH5 alpha cells. For instance, if I go back to these slides, so this is when we do the transformation of competent cells, DH5 alpha cells. We pass that through heat shock treatment, inoculated that in LB broth, and then incubated them, all those bacterial culture at 37 degrees centigrade for 45 minutes, and then spread bacterial colonies on agar plate. And then the next morning, what we have done in that we picked well isolated colony and then grow that in LV broth to make a bacterial culture. So up till this point, these all steps, they will stay the same in the transformation of protein expression strain of the bacteria. However, in this particular step, where we pick well isolated colonies and then grow them in LB broth to make a bacterial culture. So in a protein expression step, we collect 5 or even more than that, 10. So we collect many colonies, those colonies of bacterial strain. They are well isolated colonies of bacterial strain, and grow them separately in LB broth. I will show you in a moment why we do that. So coming back to the to our routine, to our slide. Now I have mentioned here that small scale screening for high expressor. So I mentioned lysis. So lysis means that if we have those bacterial cells, we have to break them by different procedures. For instance, we can use lysis buffer, we can use like sonicators or sonication, through sonication, lysis buffer, centrifugation, different techniques. It's a stepwise procedure. So that's how we extract these inclusion bodies. Now, when we are going to make a protein, and if we are interested in to make a 10-liter protein, so it's a sort of a scale-up process. Before going to a scale-up process where we want to synthesize protein, a total amount of 10-liter or 20-liter or many more than that, then we have to screen those bacterial strains which are capable or which are high expressors. So meaning that we have to use that particular bacterial colony that is good in expressing our protein of interest. So that's why we do this small scale screening for high expressors. So once we identify these high expressors, then we use that particular colony for a scale-up process. So the steps I mentioned before, that we do lysis of the bacterial cells so that we can extract the inclusion bodies inside the bacterial cells. We do that by lysis and then followed by washes, buffer washes, and then centrifugation. So it's a very lengthy procedure, but I didn't mention it here. And the ultimate goal is to collect inclusion bodies. So once we have these inclusion bodies, as I mentioned before, that inclusion bodies, they're in an aggregated form, so we have to unfold them into a primary amino acid linear structure, because they're not folded. So we have to refold them into a complete 3-dimensional structures. But before that, we have to unfold that, and this unfolding takes place in a highly denaturing environment. For that, we use 7 molar guanidine HCl or 8 molar urea. So once we unfold that protein, and then we have to refold that in a refold buffer. I would just mention a volume of 10 milliliter just to give you an idea that we started. For instance, if we start with unfolding, that's about 10 milliliter volume, and then we do a refold. So refold is a bit large volume, for instance 2 liter or 5 liter, because the idea is that in a refold buffer we want to give a very relaxed environment for a protein molecule so that they can refold properly in a refold buffer. Okay, so now how do we do the refolding of unfolded protein? Let me draw a figure for that. So if we have this beaker and we have around 2-liter refold buffer, and we have this small tube or a flask. So we use sorry, not flask, the falcon tubes. Now we use small tubes, and through these small tubes we put the unfolded proteins in the form of tiny droplets into the refold buffer with continuous stirring, and continue this whole process, this dripping of unfolded protein into a refold protein until all the unfolded protein gets into the refold buffer. So we can then leave this refold buffer having this unfolded protein for a few hours. Okay, so that's how we do the refolding. Now, what do we have in a refold buffer? We have a number of chemical constituents in that. I have mentioned just a few of them. For instance, we have L-arginine monohydrate chloride. It is used to prevent protein aggregation. We use tris to maintain a particular pH. We use salt, NaCl, that increases the ionic strength, and it also helps in the solubility of the protein. So these are some of the main constituents of the refold buffer. However, the constituents, it differs among different labs for depending upon the nature of the protein, and then that's how we refold a protein. So you can see here that I have used I have mentioned 2 liters. So we started with 10 milliliter of the unfolded protein, and we refolded that into a 2-liter refold buffer. So that's how we started with a very small volume, which is 10 mil, and we refolded that in 2 liter of a refold buffer. And the basic idea is that, as I mentioned before, that we have to provide a very relaxing environment for these unfolded proteins so that they can refold that properly in this refold buffer. Okay, so the next step is to concentrate that 2 liter into a required volume depending upon the need of the researcher, because like we started with 10 mil unfold buffer and we refolded it in 2 liter, which is a very diluted form of the protein, and we need a protein in a concentrated form in order to do different types of experiments with that. So for instance, if you want to concentrate that into sorry, my drawing and writing is not that much good in this. So if we want to concentrate that into to a 50 milliliter, for that we use a particular equipment that is known as the TFF, tangential flow filtration system. So it is one of the system that we use for concentrating a particular protein sample. So how does it work? The sample is passed through small pore size membranes installed in this system, which retains proteins while water and other chemicals are constituent, they will come out. So our protein of interest will be retained inside the system, which we will elute later, but the water molecules and the other constituents, chemical constituent, it will come out, so we can then discard that. And that's how we do, we use this tangential flow filtration system to concentrate our protein of interest from that 2 liter sample into a particular volume. Okay, so the next step is the purification. So it's that step. It is the last step. I mean, the aim of the process, the procedure was to purify a pure form of our protein of interest, so that we can use it for different purposes. However, based on the properties of the protein, we select different purification techniques. For instance, we can purify our protein by using affinity chromatography. That means that we should have a certain tags attached to our protein of interest. For example, 6-histidine molecule. I will talk about that in detail, how this procedure works. But if we have 6-histidine, a patch of 6-histidine amino acid residues either at N terminus or the C terminus of our protein of interest, we can use that as an affinity tag and purify our protein through affinity chromatography. We can also go for size exclusion chromatography. So based on the size, we can also purify our protein of interest, and we can also go for ion exchange chromatography, which is basically based on the charges on the protein. So we have to define or determine the charge on our protein of interest, and based on that charge we then select a particular ion exchange chromatography to purify our protein of interest. So that's all for today's lecture. If you have any question, please ask in the comment section. I will try to answer them. Thank you very much.